Welcome to MycoTalks. Today we will be hearing two talks from Anne Puel and Michelle Momany. And the organizing committee of the MycoTalks is listed here below on the slide, and it's sponsored by the MRCC in medical mycology at University of Exeter and the UKRI. The philosophy and origin of the MycoTalks goes back to the pandemic, when we decided to do online talks in a webinar format. And the philosophy is that we like to promote diversity of career stage, gender, geography, and academic field. And each time we offer two 30-minute talks on all aspects of medical mycology for the research community. And then following those two talks, we have 30 minutes of in-depth discussion. If you have questions, please submit them through the Q&A. They'll be seen only by the moderators, and it may be that some talks will be, some questions will be consolidated in the name of time. However, we usually send the questions to the speakers afterwards so that they can contact any person who's asked a question who didn't get it answered. Also note the name of the speaker for whom the question is that you're writing. Recordings of these talks will be available on the MRC CMM YouTube channel, and you're welcome to go there. These should be posted by sometime tomorrow. We have upcoming MycoTalks from in June from Mari Nover and Xin Lin, and in July from Jane Coles and Christina Cuomo. And finally, for trainees, there's a second series in the Medical Mycology Trainee Seminar Series, which features students, postdocs, clinical trainees, and new faculty. And you can present your work there to our global community. Those talks are on the second Thursday of the month and can be the timing can be switched depending on the geographic time zone of the speaker. So if you're interested, there's a link here, and I'm sure that one will be provided in the chat very soon, and you should contact the organizers if you're interested in giving a talk. And now, Neil Gao will introduce our first speaker.
Thank you very much, everybody, and a very, very warm welcome to Anne Puel, who's our first speaker today. Anne is a real scientist of the world who has addresses in both Paris and in Rockefeller, New York. She is officially Director of Research and INSERM and an adjunct senior researcher at the Laboratory of Human Genetics of Infectious Disease at the Imagine Institute Necker Hospital for Sick Children, University of Paris, and also St. Giles Laboratory for Human Genetics of Infectious Diseases at Rockefeller University in New York. And she's a group leader for human genetics of fungal diseases. I'm sure you all know that she has a real international reputation for understanding genetic susceptibility to fungal diseases and indeed other diseases as well, affected by SNPs and mutations in various genetic sensing pathways. And she's done some incredibly important work recently on immunology related to IL-17 and also on autoantibodies. So I don't want to cut too much time into your presentation. Anne, you're very, very welcome. Delighted to have you speaking to MycoTalks. And over to you to take us through your presentation today.
So thank you so much, Neil, for this very kind invitation, and it's real a great pleasure, and I would like to thank the organizer for giving me the opportunity to present the work we've been doing over the last almost 15 years on human inborn errors of IL-17 immunity in underlying chronic mucocutaneous candidiasis in humans. So as a general introduction, we all are in contact with microbes. However, only a minority of us will develop an infectious disease. And whereas most of us remain asymptomatic or will develop a benign disease, which is a rule, the severe disease is an exception. So we're trying to understand what accounts for this huge variability among individuals in the course of an infection. And we try to identify which are the crucial mechanisms which are involved in fighting against those infections. So of course, the microbes play a role, because we need to develop an infection, we need to be in contact with the microbes. And both environmental and host factors play in the course of the development of this disease. And within the lab, we are mostly involved in the study of immunological factors, such as autoantibodies against cytokine, or genetic factors which impact the response to an infection. So in this context, I'm going to talk about chronic mucocutaneous candidiasis, which is a disease characterized by severe, persistent, recurrent infection of the nail, the skin, and the mucosa by Candida species, and mostly Candida albicans. So chronic mucocutaneous candidiasis is quite frequent and associated with many other clinical manifestations in patients with acquired conditions, such as patients infected with HIV and AIDS. And some patients also under immunosuppression or steroids may also have mucocutaneous candidiasis. Among primary immunodeficiency, patients with T-cell disorders, such as severe combined immunodeficiency, in which patients do not have T cells, or combined immunodeficiency, in which patients have low T cell counts or normal T cell count, but impaired T cell function. These patients, almost all of them, are vulnerable to chronic mucocutaneous candidiasis, suggesting that T cell immunity is critical in fighting against CMC. There are also a number of other primary immunodeficiency in which chronic mucocutaneous candidiasis is one of the main clinical manifestations, and there are also few other clinical manifestations. And in these PIDs, primary immunodeficiency, we didn't see any broad T cell defect as here, and we were wondering why these patients were susceptible to chronic mucocutaneous candidiasis. So these primary immunodeficiencies are autosomal dominant hyper IgE syndrome, which are caused by dominant negative heterozygous mutation of transcription factor STAT3, patient with Cas9 deficiency, which is an adapter which is mainly expressed in myeloid cells and which help the signaling downstream of C-type lectin receptors, patient with autoimmune polyendocrinopathy syndrome type 1, which are highly vulnerable to autoimmune disease, suffer from chronic mucocutaneous candidiasis as one of the main infectious disease early in life. And last, some patients with IL-12 beta 1 deficiency or IL-12 P40 deficiency who are susceptible to a mycobacterial disease. Around 25% of these patients will also develop chronic mucocutaneous candidiasis. And when we started to study patients with chronic mucocutaneous candidiasis, we knew that there was another group of patients with what we called idiopathic CMC, because we didn't know the cause of the CMC in those patients, except that CMC was usually developing early in life. It could be either isolated, meaning that the patient did not display any other infectious disease, or syndromic. It could be associated with other clinical manifestations, but we didn't know the cause. In the early 70s, a number of multiplex families with several cases within the families of consanguineous families with chronic mucocutaneous have been reported, suggesting that there was a genetic cause for CMC. However, for almost 40 years, we couldn't find any genetic defect responsible for chronic mucocutaneous candidiasis. And it's the progressive description of these primary immunodeficiencies and the identification of impaired IL-17 mediated immunity in these primary immunodeficiencies in parallel to the work which has been done in mice, mostly by Sarah Gaffen's group, that it suggested that this study paved the way for the identification of a large number of genetic defects associated with either isolated or syndromic CMC. All these genetic defects are shown either in blue, meaning that the blue genetic, the mutation in gene shown in blue, encoding protein shown in blue, are associated, were identified in patients displaying isolated chronic mucocutaneous candidiasis, whereas mutation found in genes encoding the protein shown in red were found in mutation displaying syndromic CMC, meaning chronic mucocutaneous candidiasis associated with other clinical features. As you can see here, the description of patients with inborn errors of IL-17F, IL-17RA, RC, and ACT1, all these patients having low or abolished responses to IL-17A and F, demonstrated that IL-17A and F in human plays a major role in mucocutaneous protection against candidiasis. So, and today I'm going to present you two stories we have recently reported on other genetic defects responsible for chronic mucocutaneous candidiasis. So the first one is about this multiplex family who was reported in the lab because early chronic mucocutaneous candidiasis, which was associated with a developmental disorder. When we performed the immunophenotyping of this patient has a normal basic immunophenotyping, so we didn't know what was going on. So we performed whole exome sequencing in the 3 patients, and by applying a number of filters, we ended up by identifying a heterozygous variant of MAP kinase 8 in those 3 patients, which was common to the 3 patients. And this MAP kinase 8 gene was pretty close to the core genes of IL-17 pathway. The mutation was private, meaning that it was not present in any in the public database. It was predicted to be deleterious, and MAP kinase 8 is one of the most conserved genes in the human population. So this suggested that this mutation could be disease-causing. So indeed, we performed the familial segregation, and the 3 patients carried the mutation. And this mutation was affecting an essential splicing site of MAP kinase 8, which encode the 4 isoform of JNK1. So in an overexpression, in an in vitro exon trapping assay, we showed that this variant was associated with aberrant splicing mRNA with either intron 4 retention or exon 4 skipping, both of them leading to premature stop codon protein. And when we overexpress this mRNA in HEC293 T cells, we can see that these two predicted mRNA couldn't encode any JNK1, were complete loss of expression. So this was in an overexpression system. And what about the patient cells, which are heterozygous for the mutation? So we looked at immortalized B cells or immortalized SV40 fibroblasts, so from the skin. And again, in the patient, as compared to the control, we could see aberrant mRNAs. And when we look at JNK1 protein expression in the patient cells, EBV cells and SV40 fibroblasts, we could see that this mutation was associated with a strong reduction of JNK1 protein expression. So JNK1 is involved in many signaling pathways, and in particular downstream of the IL-17 receptor, downstream of IL-17A and F. And in particular, upon IL-17 receptor stimulation, it phosphorylates C-Jun and ATF2. So we look at C-Jun phosphorylation and ATF2 phosphorylation upon a time course of stimulation with IL-17A in the patient as compared to 2LC control or a patient with a complete IL-17RA deficiency. And as expected, we observed an impaired phosphorylation of C-Jun or ATF2 as compared to control cells, and almost comparable to what we saw in the IL-17RA deficiency. We also looked at the transcriptomic profile in transcriptomic profile upon a time course of IL-17 stimulation in the two patients as compared to 2LC control and a patient with complete IL-17RA or IL-17RC deficiency. And again, as you can see, there was a strong impairment of the upregulation of a number of genes in the patient or the downregulation of IL-17 dependent gene in the patient to levels almost comparable to the IL-17RA or RC deficient patient. And here I'm showing you some of the gene which have been impaired, which induction was impaired in the two patients as compared to the LC control or as compared to patients with IL-17RA or RC deficiency. And last, we looked at the induction of IL-6 or Gro alpha in the patient cells. And again, we stimulate the patient SV40 fibroblast with increasing doses of IL-17A, IL-17F, and IL-17AF as compared to LC control in blue or a patient with complete IL-17RA deficiency in purple. And as you can see here, heterozygous cells of the patient showed a strong impairment of Gro alpha or IL-6 induction upon IL-17 stimulation. And to demonstrate that this was due to the impaired expression of JNK1, we transduced the patient cells with cDNAs encoding the 4 isoforms of JNK1, and as compared to the cells transduced with an impaired, with an empty vector upon stimulation with IL-17A, we could see that the transduction of wild-type MAP kinase 8 restored the production of Gro alpha and IL-6, demonstrating that the impaired production of the impaired responses to IL-17A and F in those patients was indeed due to MAP kinase 8 or JNK1 deficiency. So up to now, we have now identified 5 genetic defects which either impair or abolish the response to IL-17A and F, and all the patients are susceptible to chronic mucocutaneous candidiasis. So IL-17A and F are critical in protecting against mucocutaneous candidiasis. Some of these patients also display Staphylococcus aureus mucocutaneous infection, so suggesting that it might also play some role to protect against this infection. However, none of these patients up to now, so approximately altogether we have around 40 patients, which is not a lot, but it's still something. None of these patients display other severe infections, suggesting that IL-17A and F plays a redundant role against in the protection against most of the other microbes. So the second story is about this young girl that we recruited in the lab, again for a number of infectious diseases, and as you can see here, severe oral and esophageal candidiasis. So when she had also additional infectious disease, but when we look at the immunophenotyping, it was globally normal, except that the patient had low NK cell count, and she wasn't able to produce antibodies following vaccination with protein antigen or polysaccharides. And she also showed a strongly profound impaired cell proliferation to antigen in vitro. So again, we performed whole exome sequencing, and we ended up by identifying a homozygous private variant of REL. And this variant was supposed to be highly deleterious No other homozygous predicted loss of function mutation of CRL were identified in the databases. And when we perform the familial segregation, so only the patient who was sick was homozygous for the variation, whereas the LC parents and the brother were either wild type or heterozygous for the mutation. So this variant was in the REL gene, which is called CREL, which is one of the 5 transcription factors of the NF-kappa signaling pathway. And again, it was supposed to affect an essential splicing site. And as expected, so this mutation was associated with a deletion of 14 base pair, leading to a frame shift and the early premature stop codon. When we looked at the CREL or REL mRNA and CREL expression in the patient B cells, the mRNA was barely detectable. We couldn't detect any CREL protein expression by Western blot or by flow cytometry in the patient cells, and in an overexpression system to assess the function of this allele in HEC293 T cells, which were transfected with either with wild type or mutant CREL and an NF-kappa-B luciferase reporter gene, we could see that whereas the wild type allele was able to induce NF-kappa-B activity, the mutant protein couldn't, demonstrating that this allele was completely loss of function. So CREL, as I said, is one of the 5 transcription factors of the NF-kappa-B signaling pathway. It belongs to the canonical pathway, and it's mostly expressed in hematopoietic stem cells. And it's activated by various receptors, such as T cell receptor and B cell receptors, toll-like receptor or cytokine receptor. So because the patient has, so we first look at the different cell population in the patient, and we found that the patient has globally a normal proportion of most lymphoid cells, except that she has low memory CD4 T cells, low MRA CD8 T cells, low proportion of Th1 and Th1 star. When we were looking at the proportion of Th17 cells as assessed by extracellular staining of receptor, she was in the low range of the control, but she had some proportion of Th17 cells. She had also low Treg cells and low memory B cells. So as I said, CREL is involved in a number of functions, and in particular in cell proliferation. So we looked at naive CD4 T cells, we purified naive CD4 T cells and looked at naive CD4 T cell proliferation upon TH0, which TH0 meaning stimulation with anti-CD2, CD3, and CD28. And as you can see, as compared to control cell, the patient had a lower proportion, an impaired proliferation upon TH0 condition. And under this condition, she barely was able to produce IL-2. So probably the impaired IL-2 production by naive CD4 T cells was associated with the impaired proliferation and possibly impaired T cell survival. We also assessed the naive T cell proliferation under this TH2 condition, TH0 condition by adding exogenous IL-2. And indeed, we were able to restore, to improve the proliferation as compared to in the absence of IL-2. However, we never get to the cells never reach the proliferation level of LC individual. And this impaired proliferation and probably survival was associated with a strongly impaired production of cytokine. And in particular, we looked at here at the IL-17 cytokine produced by naive CD4 T cells under polarizing condition during 5 days. And as you can see here, in contrast to control cells, the patient barely produced any IL-17A, and the IL-17F production was strongly reduced. And when we look again at also at the production of IL-17 cytokine by memory CD4 T cells by 5 days of stimulation by anti-CD2, 3, and 28 beads, the patient again display very low production of IL-17 cytokine. So this impaired IL-17 cytokine production probably explained the chronic mucocutaneous candidiasis in this patient. And very recently, we identified 2 patients with a defect not in CREL, but in REL-B, which is another transcription factor of the NF-kappa B signaling pathway, but not belonging to the canonical pathway, but the non-canonical pathway. The 2 patients display a severe chronic mucocutaneous candidiasis. And in those patients also, we saw an impaired, possibly proliferation of the T cells. And most importantly, we could barely detect any IL-17 cytokine. So this again suggests that IL-17 is important. And last, I'm just going to finish with a I'm just checking the time, with some unpublished results. So in 2018, we reported a multiplex family with 2 cases with 2 patients suffering from severe mycobacterial disease and autosomal recessive IL-23 receptor deficiency. So this patient had a complete loss of response to IL-23. And in contrast to what was expected from our studies, we asked and we asked and we asked, the patient never displayed any susceptibility to chronic mucocutaneous candidiasis. So more recently, we recruited 4 additional patients with mycobacterial disease, and 2 of them display chronic mucocutaneous candidiasis. And by whole exome sequencing, we found that all these patients display a mutation, homozygous mutation in IL-23 receptor. So in contrast to the first family published, at least 2 patients display chronic mucocutaneous candidiasis. When we looked at the expression of IL-23 receptor in EBV cells transduced with either the wild type, the mutation previously identified, or the patient mutation, we found that this mutation were associated with either abolished expression of IL-23 receptor or with normal or reduced IL-23 receptor protein expression. However, when we looked at a STAT3 phosphorylation upon IL-23 stimulation, we can say that all the mutation, we didn't detect any STAT3 phosphorylation, demonstrating that this mutation were complete loss of function. So we looked at the proportion of Th17 cells ex vivo, and as previously reported, when we looked at the proportion of Th17 ex vivo by extracellular staining of extracellular receptors, the proportion of Th17 cells were comparable to the control. However, when we looked at the proportion of IL-17 positive among CD4 T cells, the patients where we barely were able to induce any an IL-17A induction, except upon PMA ionomycin, but still significantly at levels significantly lower as compared to the control. And we could see that upon IL-23 stimulation, mate cells were able to slightly induce IL-17 and IL-17 expression, and as compared to the cells which were not stimulated, but the patient did not induce the patient cells did not induce any IL-17, nor upon PMA ionomycin, we couldn't detect any IL-17 induction. And last, when we looked at the production of IL-17A and F in PBMC of the patient stimulated with heat-kill Candida albicans, we couldn't detect any IL-17A or any IL-17F production, in contrast to the control cells. So suggesting that all the patients, despite they suffer or not from chronic mucocutaneous candidiasis, they have an impaired production of IL-17 cytokine upon Candida albicans infection. So in conclusion, We have now, since over the last 12 or more years, we have identified 19 additional genetic causes responsible for either isolated or syndromic CMC. As you can see, all the genetic defects we have identified up to now either impair or abolish the response to IL-17AF, abolish the response to IL-17AF or the production of IL-17AF, demonstrating that these two cytokines are critical in mucocutaneous protection against Candida albicans. Finally, I would like to thank, of course, all the agencies who are supporting our work. All the patients and the families who are participating to the studies, our lab of human genetics of infectious disease. And in particular, I would like to thank Juan Lee, who has performed the Junquan study, Romain Levy, who has performed the Cereal study, and Quentin Philippot, who is working on IL-23 receptor deficiency. All the folks, so the PhD student and the postdoc in the lab and the researcher who are working on helping on this project, and of course, all the collaborators, thanks to we have been able to do these studies. Thank you so much. Thank you.
Thank you very much, Anne. And I'll remind everyone again that we'll be taking questions at the end of the second talk, but if you have questions for Anne, please feel free to post them in the Q&A section. So now I'm going to introduce Michelle Momany, who's going to be giving us the next talk. Michelle earned her PhD in medical mycology with Paul Paul Stanislow at University of Texas, Austin, and her postdoc with John Hammer in fungal genetics at Purdue University. She's been a central figure in the fungal community, for example, a founding member and chair of the Aspergillus Genomics Research Policy Committee, a member of the Fungal Genetics Policy Committee, and the Fungi DB Advisory Board. She chaired the Gordon Research Conference on Cellular and Molecular Fungal Biology and the Fungal Genetics Conference at Asilomar. She's an American Association for the Advancement of Science Fellow, a Mycological Society of America Fellow, a National Academies Education Fellow in the Life Sciences, and a University of Georgia Women's Leadership Fellow. At the University of Georgia, she's been a department head for plant biology and is currently an associate dean in the Franklin College of Arts and Sciences. Research in the Momany lab focuses on two general areas, basic biology and antifungal resistance in the pathogen Aspergillus fumigatus. Her lab characterized early development in Aspergillus, and recently her lab has focused on understanding dormancy and germination of Aspergillus fumigatus spores and on antifungal resistant and Aspergillus fumigatus in the environment. Other research has focused on cell polarity and the septin cytoskeleton in the model system Aspergillus nidulans, with a recent focus on the evolution of the septins and interactions among the septins. So welcome, Michelle. We're looking forward to your talk.
Thank you, Judy. Thank you, Judy, for that gracious introduction, and everyone for bearing with the changing up all the screens here. It's my pleasure to tell you today about two projects going on in my lab. In the first one, I will be talking about Conidial dormancy and germination, and in the second, about azole resistance. So for the first project I'll be telling you about, we'll be talking about Canadia, and of course, Canadia are dormant spores. Here I show a conidiophore of Aspergillus nidulans, and you can see these spores, the conidia, in a chain coming out of this specialized structure. In Aspergilli, these conidia have a single nucleus, so they're uninucleate. Of course, all spores are dormant, but in this case, the dormant spores are asexual. So these are the product of mitosis. So if you started with a single conidium in this colony, everything on this conidiophore and actually everything in the colony should be identical. They're all products of mitosis. Conidia are also abundant. A single colony makes millions of spores. They're very small. So Aspergillus fumigatus conidia are between 2 and 3 microns. That means that they're very easy to disperse by wind and also very easy to inhale, which is how the disease is acquired. Now, for medical mycology, this is very important because if you look at the most common invasive fungal infections, the majority of them start with spores. Obviously, Candida albicans is the big exception there. But if you look at this list with Aspergillus fumigatus at the top, all of these fungi make spores. Most of them are asexual spores or conidia that can then be inhaled or land somehow in the host and cause life-threatening diseases. So one of the things that makes conidia very good at causing disease is that they are what we call phenotypically plastic. And by that I mean that the conidia are able to survive in different environments to sort of adapt to the environment. So my lab got interested in the question of how does this phenotypic plasticity occur? What underlies that? If you think about it with sporulation, which is this production of spores, and germination, which is the breaking of dormancy and then switching to vegetative growth, in the case of filamentous fungi, then extending their hyphae, invading the substrate, that sporulation and germination can occur in very different environments and usually do occur in very different environments. So if we look at human pathogenic fungi, again, well over 1 million invasive fungal infections a year are thought to be caused by fungi where spores being inhaled is part of what starts that infection. So if you look at sporulation in these cases, many of these fungi are saprobes. They live in the soil or organic litter, and that's where the sporulation can occur, these conidia being made. But the germination, at least in the case of Fumigatus and similar pathogenic fungi, occurs in the human lung, which is a very different environment. So the fungus basically has to be formed in one environment and thrive in another. If we look at plant pathogenic fungi, there are probably billions of disease cases a year if we want to think of each leaf as a patient. And that sporulation also can occur in a very different environment than germination. The germination part is on a leaf, a root, a stem, depending on the specifics of the fungal disease. So with plant pathogenic fungi, many of you may be aware, it's something like 20% of crops every year that are destroyed by plant pathogens. So what enables this phenotypic plasticity of the spores, allows them to be made in one environment and thrive in another? One way to think about this is there a single germination program, or are there multiple programs within a spore? So for example, are all the spores that are identical from a colony programmed in the same way, and then when they land somewhere, they adapt? Or are they programmed in subtly different ways while still on the conidiophore, and then only some survive in the new environment? So another way to phrase that is the question of, does that sporulation environment determine what we call the germination potential, the ability to break dormancy and thrive in a new environment? So to answer that question, we set about doing some experiments. I'm going to point out at the beginning of this work has recently been published in the Fungal Genetics and Genomics collection of G3, which is published by the Genetic Society of America. So if you want to get more detail, that's where you can go to find it. So what we did to answer this question was a series of sporulation germination swaps. And the way that worked was we took Fumigatus, sporulated it on solid medium for 72 hours, washed, and then germinated in liquid medium for 6 hours. That was followed by fixation and then flow cytometry to look at the size of those Canidia and early germlings. So we're using size here as a proxy for breaking dormancy. So as I mentioned, we started with solid medium sporulation for 72 hours. And in this case, I'm showing you complete medium. Those spores then would be harvested and identical aliquots taken to these 9 conditions. And you can see there, some of these are environmentally important conditions. Some are known to be medically important. For example, low iron, Ross stress, high temperature is one as well. So after the complete, we also took spores from minimal medium and then introduced aliquots, identical aliquots, into the same 9 conditions, 50 degrees into the same 9 conditions, and so on. So that ultimately what we had was a complete 9 by 9 grid, so each of the beginning conditions to all 9 for germination. And that was repeated 4 independent replicates, and we got very similar patterns each time. So just to walk you through how to look at this data for what I'll show you in a moment, what I'm showing you is the sporulation in all cases on these histograms will be shown in the pink or red color. Germination, which is after 6 hours in the liquid medium, is shown in blue. So here, the pink peak shows you the forward scatter of the dormant spore population. The blue peak shows you the forward scatter of the population after 6 hours. And in this case, we're going from complete medium for sporulation to complete for germination. And just to show you, we've done microscopic observations. I've shown you the morphology of the cell up here along the top. So the pink part, which is the dormant Canidia, as I mentioned, are about 2.5 microns. And then as the germ tube emerges, you can see where that forward scatter is. In complete medium, by the time we get to 6 hours, you can see that the vast majority of these Canidia now have broken dormancy and very synchronously progressed to this young germling stage. So I'll show you just another result here. This one's not terribly surprising. If you look at the sporulation condition on this axis, here's complete sporulation, minimal sporulation, and now germination incomplete and minimal. What you'll see here, if you remember the size here, in complete medium, when it's sporulated and complete, most of the germlings reach, most of the Canidia reach this germling size, and that's whether you've come from complete medium for sporulation or from minimal. And similarly, if you look with minimal now, you'll see that they are moving in a fairly synchronous manner, but most of the germlings are quite short, whether you've started with complete or minimal after that 6 hours. Now I'm going to show you the whole 9 by 9 grid. It's a lot to look at at once. The sporulation conditions run along the y-axis, the germination conditions along the x-axis, and if you want to dig into your favorite conditions, again, there's the reference there in the corner for you. What you can see just by looking at all of these at once is that, you know, the dormant Canidia look fairly similar in everything. The peaks, you can see there's a pattern to them as you go down the columns and across the rows. So let's dig a little deeper. First, if you look at the 50-degree sporulation, so that's here, I'm showing with an arrow. In 50-degree sporulation, notice that that red peak is shifted ever so slightly to the right. Since we're tracking forward scatter, and that's our proxy for size, what that means is that the Canidia produced at 50 degrees C are slightly larger than those produced at 37, which is our standard for every other condition. And indeed, when we went in and looked microscopically, the 50 degree are slightly larger. It's about 1.5 times larger than those made at 37 degrees. Another one of the more striking results is if we looked at sporulation on limited zinc and then looked at the germination. What you see as you move across this row with the sporulation into different germination conditions, first is that not as many of the Canidia are breaking dormancy and growing, so they're not extending as much. The other thing that was quite striking is instead of getting those tight peaks that I showed you earlier, we're now getting much more broad peaks or not much movement at all off of that size of the dormant Canidia. So to remind you, as you move from left to right along the x-axis, what you're seeing is the size of those swelling Canidia and early germlings. So what this means is that the limited zinc sporulation not only reduces the ability to germinate, but it also reduces the synchrony of that population, putting more variation in, because we're seeing a lot more of all of the sizes represented. So I've shown you then that the sporulation conditions do influence germination. The next thing we wanted to know is if that sporulation environment influenced virulence. And to look at that, we used the Valeria model. And I'm just showing you, again, a subset of the results. But what you'll see across the top row is what the sporulation conditions were. So I'm showing you here minimal medium, which is sort of our control at 37, low zinc medium, and 50 degrees, so high temperature. The images of the Valeria in the next row are showing 36 hours after injection of the Aspergillus fumigatus spores that were raised on the condition shown at the top. And with Valeria, the way that you see death of the Valeria is when they turn black. So I think you can see pretty clearly here that the 50 degree, you have a lot more black of the Valeria here than you do with the minimal or the zinc. We also did some statistical analysis across multiple experiments, a total of 75 individuals for each experiment, and calculated the survival time, the average survival time. And what we found is that in minimal medium, it's a 31-hour survival time, but in the 50 degree, it's 22-hour survival time. So these Valeria are dying much more rapidly from Canidia that are raised at 50 degrees. So I've shown you then that the sporulation environment influences the ability to germinate and the synchrony of that germination, that the sporulation environment influences virulence, at least in a Valeria model, and that this Canidia sporulated under the same conditions can be somewhat heterogeneous. In the case of the low zinc medium, they're very heterogeneous. And again, if you want to look at your favorite conditions in a little more detail, the publication is out there. So the other thing we wanted to know then is that sporulation environment influencing the transcriptome? Can we pick up the changes that we saw in phenotype at that level? And this is work that was done by Earl Kung, who is the graduate student who actually was the first author on the work I just showed you as well. He funded this using an interesting platform that's called experiment.com, where you can crowdsource experiments. So this RNA-seq was done through crowdsourcing. And what Earl did was take the conditions shown here, raise the Canidia under these conditions, so sporulated them minimal, 50, low iron, high salt, or low zinc, and then did RNA-seq on the dormant Canidia. And I'm going to kind of summarize those results. This is a very tortured Venn diagram, but what you can see pretty clearly here, there is a set of 4,400 transcripts that are found across all of the conditions, so all of the dormant spores have those. And then each of the conditions themselves has some unique transcripts. Minimal medium has 27 of them, and then as you might expect from the phenotypes that I showed you, 50 degree has a higher number, so 182, low zinc, 207. So the 50 degree and the low zinc both have more unique transcripts from having been sporulated on that condition. Now, while this work was in review, my colleague Chris Wong at University of Macau did some really lovely studies following up on this idea further. Remember that when you look at the Canidia 4, the chains of Canidia are coming off of it. So the newest Canidium is at the bottom of the chain, and then as more Canidia are made, the new one comes in and pushes that chain upward. Okay, so the youngest one is at the base. What Chris's lab did was the same set of sporulation germination swaps that I just showed you, that Chris and his lab had seen this, had talked to me about this at a previous meeting. And they did those sporulation germination swaps in Aspergillus nidulans, Fumigatus, and Alaromyces. What they showed was that as long as these Canidia are attached to the Canidia 4, the transcripts are altered by the medium. So this transcriptional changes occur even after the Canidium's fully formed, as long as it's attached. So if you shift out the medium in this condition, you're still getting transcripts changing in response to that medium. And of course, I'm just summarizing the main finding. This is a really beautiful paper. If you haven't seen it yet, it's well worth a read. So what I've shown you then is that the sporulation environment does influence the ability to germinate. Canidia sporulated under the same conditions are heterogeneous, and that we know that the transcripts within those Canidia are responding to the environment. So what this suggested to us, and sort of the working hypothesis we have right now, is that this suggests that Aspergillus fumigatus is using a form of bet hedging. So bet hedging is when an organism has a lot of progeny, it builds in variation in those progeny, so that no matter what environment they find themselves in, some of them can survive. I would also say part of our kind of working hypothesis right now is that this is probably seen in other conidial fungi. If you think about fungi and Canadia, there are so many progeny coming out. If they're windborne, they end up going to lots of different environments. So my suspicion is that this kind of built-in heterogeneity is probably much more widespread than just Aspergillus, probably pretty common around among those fungi that have windborne Canidia. Now with that, I'm going to use the rest of the time to shift gears a little bit to a different story. This is looking at azole resistance. And I want to first say this is a collaboration with my colleague Mirren Talbot-Brewer, who is also in the fungal biology group here at Georgia. She is a plant pathologist and really excellent mycologist. This has recently been published again in the Fungal Genetics and Genomics collection at G3. So if you want to find out more than I have time to tell you today, that's where to look. There is also a connection to the first part of my talk, though it may be slightly tenuous, and that's that we're going to be talking now about Aspergillus and aspergillosis, which is acquired via inhaling Canidia. Remember, these Canidia are windborne and can travel. As most of you know, azoles are the first-line drug against Aspergillus fumigatus, so aspergillosis, and also many other human pathogenic fungi. And it's been appreciated for a while now that the agricultural use of, that the fungal species with reported azole resistance are increasing. So what you see here, this is from a nice review that Matt Fisher did several years back. You're seeing the number of fungal pathogens of plants and fungal pathogens of humans that have azole resistance and how those have increased over the years. So but from 1997 up to 2017. As you move downwards, you'll see a lot more of the colors getting darker and a lot more areas colored in. What that means is more and more species are being identified of fungi that are either pathogens of plants in the blue or pathogens of humans in the brown that are resistant to azoles. Now, azoles target CYP51. This is a cytochrome P450 that's involved in ergosterol biosynthesis. The azole fungicides are widely used against plant pathogenic fungi. So these are used in agricultural settings, used against crops. As I mentioned earlier, fungi cause like 20% of crop loss annually in the world. Azole antifungal drugs are also widely used against human pathogenic fungi, especially in the case of aspergillosis. One other thing for those of you who don't look at a lot of the azole resistance literature, there's a specific kind of azole resistance called Panasol resistance, which means resistant to multiple azoles and often resistant to high-level azoles. And the specific mutations there are repeats, these TR repeats that are in a promoter region of CYP51A and coupled with a mutation in the reading frame. And there are a couple of them I list here that are fairly common. So it had become clear, actually it's been something like at least 10 years now, that closely related Fumigatus Panasol resistant isolates had been identified from both clinical and agricultural settings. And based on that close relationship, it was postulated that agricultural use of azoles might be the driving force for a lot of clinical azole resistance. And I list here some of the key publications in the area. You can see going back to as far as 2009 in Verwege's lab. Meis's lab has also been very active in this area. It was kind of thought that it made a lot of sense that probably azole resistance was coming from these agricultural sources. But there was still some question about how could you actually show that? Might they both clinical and agricultural azole resistance be responding to similar things in the environment other than the azole compounds? So what we set out to do, we did some surveillance of Aspergillus fumigatus in agricultural environments. I'm showing here my colleague Maren and my graduate student Earl Kong, who is all suited up to do isolation in the field. We looked at 56 agricultural sites in Georgia and Florida on a variety of crops, specifically focusing on soil, debris, and compost. We isolated 700 different isolates, but for today's talk, I'm going to focus on 12 that were pan-azole resistant. And what we wanted to know is, was there a signature of agricultural origins? My colleague Maren Brewer had just finished reading this book by Maren McKenna called Big Chicken. And in Big Chicken, they basically go through the story of how it was shown that antibiotic use in agricultural animals was leading to antibiotic resistance in clinical human populations. And so, inspired by that, Maren got the idea that what we should do is scan our environmental isolates and also clinical isolates where the genomes were available publicly for resistance to fungicides that are used only in agriculture. So fungicides that are used only in crops. And what we looked for, one of those fungicides is called QOI resistance, and the other is abinamyl resistance, and you can see the mutations there. So I'm going to jump right to the punchline, and that is that we did find clinical and environmental panazole resistant isolates that carried resistance to agricultural fungicides that are never used on human populations. So what I'm showing here is a neighbor joining tree of our environmental Aspergillus fumigatus isolates, and also a variety of environmental and clinical isolates from around the world that were publicly available. What you'll see in green, if you follow the bar around, the green shows you things that are environmental isolates. The blue shows clinical isolates, so isolates from patients. The red filled in circle shows you pan-azole resistance, so resistance to more than one azole and having this unique TR repeat combined with a point mutation that I mentioned earlier. The open red circles are azole resistance that are not pan-azole, so single azole and lower level. In gold is QOI resistance, so remember QOI is that fungicide that's only used to combat fungi that are pathogens of plants. It's only used on crops. And MBC resistance in purple. So this is the Benamel fungicide was used widely maybe 5-10 years ago. It's not used so widely now, but it was used to combat fungal plant pathogens. It's also used some against worm pathogens, so it's not strictly agricultural. But what you can see here, if you look in the blue, there are some isolates here from the Netherlands that have pan-azole resistance and fungicide resistance, and also several from India. So I've shown you then that pan-azole resistant Aspergillus fumigatus has been isolated in Georgia and Florida. Some clinical and environmental plant pan-azole resistant isolates also carry resistance to strictly agricultural fungicides, which sort of nails that they are indeed, at least this set of isolates, had an agricultural origin. There is no other way they could have picked up resistance to agricultural fungicides. I'll also mention that while this work was in review, similar results were also published from the Mulatto lab in Spain, showing kind of a similar pattern and a similar conclusion. So this isn't a one-off. It looks like it's a much wider phenomenon where we can actually now begin to track the agricultural origin of pan-azole resistance through looking for resistance to agricultural fungicides. And again, if you want more detail, obviously there's a lot more in the paper. Here's the reference for you. And then finally, I'd like to acknowledge the people who were involved in this work. In my lab, in this photo here, we were at a party in our department where you had to dress as your favorite plant or fungus. Obviously, we are Aspergillus. You can see our Canadia on our heads and hyphae skirts. Brandy Celia Sanchez here is the one that was participating in a lot of the first work I showed you. Brandon Mangum participated in the second. And then Earl Kung, who you can see over here in this picture on this side, really pushed both the projects that I was showing you and did an awful lot of that work. I'd also like to acknowledge our collaborators. Here is Maren Brewer on one of our trips to CDC, and Tina from her lab, who are both very involved in the project, and then my colleague Duda Ben Sasson in the UGA fungal group, who helped with the bioinformatics on the first project. I'd also like to acknowledge my funding sources, University of Georgia President's Interdisciplinary Seed Grants, the Center for Disease Control and Prevention, and then Maren and I are also doing collaborative work funded by the USDA and NIFA, and also experiment.com. So thank you for your attention, and with that, I'll stop for questions.
Thank you very much, Michelle. That's really fascinating. I think we'll turn it over to Neil, and he'll start with questions for Anne. And meanwhile, all of those in the audience are welcome to add more questions. Please put your questions into the Q&A section, not the chat box, because we are following things in the Q&A in order to make sure that they are all together. Neil?
And thank you very much. We have a number of questions, and I've actually managed to rescue one also from the chat box, so I'll pick up on that one. So I'm going to kick off with this question, which is actually from Gordon Brown, and he wants to know, why don't the C-REL patients with that deficiency suffer from other infections such as tuberculosis, or indeed, and do they?
So thank you, Gordon, for the question. So she suffered from other infection. She had a severe candidiasis, oral candidiasis, but she also present following BCG vaccination. She had a disseminated BCG disease with lymph nodes, bones, and severe lung disease. She had also cryptosporidiosis, and she had also other and viremia. So she had broader phenotype than the CMC, but mycobacterial disease. And she's been transplanted. So she's been transplanted, and now she's almost off of infection, but did not have tuberculosis.
Thank you, Anne, for that. Judy, do you want to alternate the questions?
That's fine. So far, fine. Yes. So
Okay.
I actually have one very short one from Bert Gerritz van den Ende, who asks if Alaromyces marneffii is the same as Talaromyces marneffii. Michelle, that's for you. And you need to unmute.
Sorry, I don't know. I'll look it up and find out.
Okay. And the second question is from Francisco Alvarez, who says, hi, this is a question. Hi, thanks for the great talk. Have you tried enzymatic treatment of the spores before the incubation and germination media to see if the sensing of the environments gets affected?
Okay, so thank you for that question, Francisco. So we haven't tried specifically that, but what I will tell you in the work that I talked about from Chris Wong's lab, he actually did look at whether ATP was being depleted, and he looked at after the Canidia have been detached, okay? So what I think is going on, he postulated, I mentioned how that chain of Canidia is the youngest ones at the bottom, there's sort of a gradient. He postulated that that might be the reason that there's variation in Canidia. I'm not so sure about that part, but I think what he did show was that as long as they're attached, you are still sensing the environment. Once they detach, that environmental sensing stops, and we get something that's more like we traditionally think of as dormancy. I'll also point out a lot of people, I think, make a little mental mistake when thinking about dormancy. Dormancy is not death, right? It's suspended animation, everything's slow, but there are still responses going on.
Okay, let me ask another one to Anne, and don't worry if they're out of order in which they came in. This one I noted when it came in the chat, but it's from Zinli, and Zinli says, first of all, thanks for the amazing talk. Question is whether or not the patient, which is the IL-23R mutation and CMC, does that patient exhibit any gastrointestinal symptoms such as inflammatory bowel disease?
So thank you. To our knowledge, no. They had they had oral esophageal candidiasis, but no gastrointestinal disease or IBD. But they're young patient, but no.
Okay. And you would probably be aware of that, wouldn't you, in talking to the patient, had it been a problem?
So we're asking, we have large questionnaires, so asking for we also ask this in the questionnaire, so we should be aware of. So, but up to now, none of the patient reported with any IBD.
Okay, thank you.
Thank you.
Okay, so for Michelle, from Elaine Bignell, who says, beautiful work. Do you know whether azole primed spores, either via lab or environmental culture, are more fit under any particular stress conditions? And secondly, do you think that spores remain transcriptionally responsive to their environments, even once dispatched from the colony? So the second one you started commenting about.
Yeah. So thank you, Elaine. as I noted in my reply to you, actually, the inspiration for a lot of this direction came from my research leave with you several years ago. So it's glad you're getting to see some of it come full circle. So we have not tested azole prime spores to see if they're more fit under any particular conditions. So this is just a guess on my part. My suspicion is because you're modifying the membrane and the membrane lipids, that there would be some change in fitness, for example, in specific high temperatures, but we have not done that experiment. And then in terms of the transcriptional responsiveness, I think I mentioned that they really, based on Chris's work, it doesn't look like they're staying responsive after they've been detached from the conidiaphore. I guess you could think of some conditions in which you might be damaging the spores so much that they can't germinate, but I don't think that's exactly what you're asking about, so no.
Okay, and now this is a question from Peter Lane, and he's asking for you to give some more information about some of the things that you've said in some of your reviews with your colleagues. So the question is, as someone who deals with APECDED, in other words, AIRE mutations, AIRE mutations, I'm familiar with their candida susceptibility, and it's strongly correlated with IL-17 and IL-22 antibodies. But in recent studies, science have tried to implicate interferon gamma in this susceptibility. And then he says, he reads in your reviews that you, in the way that you wrote these, that you interpret perhaps skeptical views about this. So what's your recent update on the relative importance of those factors?
Yes, so I guess it's regarding a paper who has been published in Science, suggesting that air deficient patients who have this chronic mucocutaneous candidiasis and who display other autoimmune features so we found together with the group of Kay Keeson, we reported in 2010 that this patient had autoantibodies against IL-17 cytokine, and that was the cause of chronic mucocutaneous candidiasis in those patients. And very recently, so last year, a paper has been published in Science saying that almost ignoring the autoantibodies against IL-17 in those patients and saying that it was due to increased signaling downstream of interferon gamma receptor. And so we comment on this article because so first of all, I was not fully convinced by the data, because the patient, yes, almost ignored the presence of autoantibodies against IL-17 cytokine in those patients, which I guess we cannot ignore it. And then they found an increased signaling downstream of, so an increased signal, STAT1 dependent signaling, possibly in interferon gamma, type 1 or type 2 interferons, we don't know exactly. But in most of the experiments which have been performed, the blocking of IL-17 immunity by this autoantibody was not assessed. And in addition, they have used a mouse model, and in mice, we don't have the presence of these autoantibodies against IL-17 cytokine. So there were a number of experiments which were not mimicking what was happening in human. So we have done this comment suggesting that I'm still not convinced that this increased interferon gamma is responsible for the CMC. And last, there are a number of primary immunodeficiency with increased interferon gamma signaling, or we can also give recombinant interferon gamma, and none of these patients were reported with chronic mucocutaneous candidiasis. So again, it's not so the patient who have received recombinant interferon gamma, it's in the adulthood or later on. So we may say, okay, it's later on. But patients with inborn errors of immunity and increased interferon gamma, they are not reported with CMC. So I'm not completely convinced about these results.
Thank you. So I think there's some appropriately correct interpretation of the way in which you worded that response.
Okay. Thank you. Okay, so from Laura Rouchet for Michelle, how much of a temperature increase is required to enhance germination of Canidia? 50 degrees is pretty warm, and might a more modest temperature increase due to climate change affect germination? And actually, what is the normal temperature that germination occurs under?
So thank you for that question, Laura. So 50 degrees is indeed pretty warm. One of the unique things about Fumigatus is it's very thermotolerant. And so there's been a lot of interest. We have another publication with Marin out about hotspots, a review of the literature where you see more azole resistance. And definitely compost, debris piles, places where you get a really high temperature, it does seem to be kind of a hot spot for development of azole resistance. Okay, so in terms of the 50 degree being, yes, it is very warm, but it's probably relevant in that context. All of the other experiments I showed you were 37. In terms of like what's normal for germination, again, Fumigatus has a really broad range, and so I think any sort of ambient kind of temperature would do. We haven't done something where we look at lower temperatures right now, but I actually do have a proposal pending to look a little more at sort of climate change kinds of changes. Based on what we've seen so far, I suspect that even a modest change in temperature is probably going to have some change in terms of this phenotypic heterogeneity, but we haven't done those experiments.
Thank you. And Anne, I wonder if I might ask quite a speculative question, which is also an ethically difficult one. But recognizing that a lot of the mutations you've identified are gain-of-function mutations, I was wondering if you would like to speculate a little bit about whether there's any relief using genome editing in the future for people who have some of these really just quite difficult lifelong genetic alterations and susceptibility, at least in theory, I should say, let's say.
So thank you. Yes. So the patient with a STAT1 gain-of-function mutation is a terrible disease, and the chronic candidiasis in those patients is terrible. So we had with Frank van der Verdonk and Bodo Grimbacher, we had applied for a grant, and we were trying actually to correct to correct this variant in vitro. And it was we didn't succeed at the time, but it was a few years ago. The problem is that again it's a heterozygous mutation, but even so we could turn off the gene because there is no haploinsufficiency of STAT1, whereas dominant negative mutation of STAT1 associated with mycobacterial disease or with mycobacterial disease, haploinsufficiency shouldn't matter. So I guess at some point we should try to do it. But because the bone marrow transplant has been used in those patients with poor success, but again, it was pretty late. So some of these patients are treated with JAK inhibitors and are improving, but still, yeah, we could. So, some days I sincerely hope that we'll be able to do it.
Yeah. Well, I hope that you're given the chance to at least try and do those in vitro experiments, because these would seem a very useful alternative way to a miserable life of long time administration of drugs, which are in some cases have got all sorts of other effects you don't want. So, Judy.
Okay, so we have a question from Nishala Nadig. She said, great talk, thank you. Have you done any chemistry work on comparing the possible toxins produced when Aspergillus fumigatus was incubated at 50 degrees versus in other media conditions before infecting Valeria with them, possibly resulting in lower survival rate in the high temperature condition?
So short answer is no. We have not done any of that work. If you look again at Chris Wong's publication that I mentioned that came out in Nature Micro, he did look at some of the toxins, and there are some environmental differences, but we have not done that.
Okay, and maybe I'll ask another one while we're here, and the question from Kim Verberg. In one of the first slides, you have shown dormant and germlings with germ tubes. Were you unable to identify the isotropic growth stadium from the other stadia using flow cytometry?
Okay, so yeah, thanks, Kim. I will tell you where this whole approach came from. Initially, I suggested to Earl, and in his early experiments, he sat at the microscope doing counts, small, enlarged germ tube. And after just one session, he came back and said there must be a better way. And he investigated using the flow cytometry. And I told him, I don't think that'll work so well, because once you have germ tubes, it's not going to work. Luckily, I was wrong. He was right, and he didn't listen to me and tried to get it going. We're very lucky at Georgia. We have a facility that allows us to put pathogens through the flow cytometry. So that helped a lot. It's from our center here that works a lot with parasites. And so they're totally comfortable with fungi. Having said that, we actually were able to look at every single morphology there. One thing I'd really like to do in future experiments, and we haven't done yet, is to actually sort based on morphology. But yeah, the isotropic, you could see pretty clearly in terms of what the forward scatter ended up looking like.
So along those lines, maybe I'll just ask one other short one, which is that it occurred to me that doing single cell sequencing on these Canidia would be really, really interesting. But I'm wondering how difficult it is to digest those cell walls and get the RNA out when you're doing it on single cells. Yeah,
so we have not yet. That's definitely a dream experiment. I would love to be able to do that. I think what might make it a little more doable, I mentioned the RNA data set that we have, and I think if we focus on a few that we know are differentially expressed depending on conditions, and then use PCR-based method to see if they're there, that might be an technically easier way, because we wouldn't need, we wouldn't be as dependent on getting the amount of material, but we've talked about various ways to do that. I agree that that's obviously the next place we should go. Technically, whether we're going to be able to or not, we'll see.
Thank you, Neil.
Thank you. Now, I've been a bit cheeky as a chairman asking Hannah questions, but I think I would like to balance out asking Michelle one, if I may. Which is, Michelle, I'm aware from my background in training in mycology that people are very interested in spores of plant pathogens on surfaces as well. And there was work showing there that the environment conditioned the spores, and one of the key factors was how old the spores were, because they basically burned up energy just to survive. And then that an old spore could be reconstituted to be more virulent by adding nutrients, and that actually could also be made more virulent by removing the competitive microflora with antibiotics. And this was sort of summarized in a concept called Inoculum Potential. And I was wondering whether in this, again, the age of the spores would affect these parameters by essentially reducing the energy which is stored and available to survive in the environments in which they then have to go and compete.
Yeah, I think for Aspergilla, you're spot on. We, again, referring to the work of my colleague Chris Wong, as I mentioned, he showed that ATP started to deplenish from these Canidia once they were detached from the conidiophore. He did do several different kinds of experiments, though, looking at age, and it doesn't look like there's a big effect of age in the kinds of timeframes he was looking at. Okay, but I do think over longer periods of time, that loss of nutrients, ATP, whatever, is probably a factor. Yeah.
So I have a question here from Paul Dyer. It says, very nice, intriguing talk. Do you think all spores are potentially viable and they just need the correct conditions to trigger their germination? Because we usually only see about 70 to 80% germination rates on standard media. So would the remaining ones just be dormant rather than non-viable? And also there's a question about the lifespan of spore survival, like months, years, but maybe too much for now.
Okay, so that's sort of a nice follow-on from Neil's question. Basically, what the one way I can't answer this, and this isn't a supplemental for that first paper, we did often get the question, especially in those low zinc conditions where you have a lot after putting them in the liquid medium that don't become any larger than the dormant spores. Are those dead? Or are they still dormant? And we did go in and do live cell imaging, propydium iodide, and one of the fluorescent trackers as well. And it turns out that most of those that are stacked up in the same size as the dormant Canidia are alive. They are not dead. So I think probably given enough time and the right conditions that they would eventually break dormancy. We also were really careful in this case to do, instead of doing a point inoculum and allowing the colony to grow out and isolate Canidia, we did a complete smear on the plate with sort of a single layer so that everything was growing at about the right time. I think some of the variation we end up seeing, like if we're not super careful about it, is because you have this difference of conidiation starting in the center of the plate. So you've got older ones there and younger ones out toward the edges. But I do think most of them, that 20% that you don't see break dormancy, it probably is conditional. And if you gave it long enough for the right conditions, it might.
Okay, great. Well, I think our time is up, and I'd like to thank all of the speakers for great presentations and the participants for good questions. Those questions that came in later and that weren't addressed will be sent to the speakers afterwards so that they can answer those who asked them individually. And next month on June 30th, you can come back to hear Mary Nover and Xin Lin. And thank you for coming.
Yeah, thanks again. Fantastic talks and wonderful to have contrasting talks, which takes us across different elements of mycology. Brilliant. Thanks very much indeed to both of you.